In this video, I'll do an example EcoToxChip analysis. So from the home page, we start by clicking here. And so this is the upload page. We'll be doing a differential expression analysis with some Japanese quail data, version 1, 384 size chip. I have these in my downloads. And so you can see here there's 15 separate individual files. From this preview, you can see that there's 2 columns, one with the well ID and then the other with the CT value. So these are what the files look like when they're created by the software that's run along with the qPCR machine. So I'll highlight all 15. And then now it's just processing it. So we click Process Files and then Proceed. Now on this page, we can change the sample names if we want to, although these are already named quite well. And then we'll put in our sample descriptors. So we have 2 doses, a high and a medium, as well as a control. So we'll put dose as our main metadata, and then we'll put control, medium, and high. And then we'll go insert. And now we can give each of the samples one of these 3 labels. So these are this H1 through H5. These are high. These next 5 are medium. And then the rest are control. It just says here a separate chemical EE2 because we had several chemicals were sharing the same controls. And then our medium and our high doses are of chlorpyrifos, the pesticide. So next we'll proceed. This next page is the quality check. So we have some tests for our housekeeping genes. This test here looks at the strand synthesis efficiency, and then this one here tests for genomic DNA control. And there will be plots down here below that show all of them. We have some default thresholds in here, but you can change these and reevaluate if you wish. We also have a view of the whole chip. So by default, it shows the average CT, but we could look at any one plate in particular. And if you click on a well, it pulls up a violin plot of the CT values across the different experimental conditions. So this can help you get an idea for inspecting the plates in more detail. So the very first quality check plot here are CT values for the housekeeping genes. We can see that in general they're okay, but it seems like this one right here might be failing. It might have a statistically significant difference across genes, but we can check by looking at the CSV plot. All right, yeah, so we have this B2M is in increased status. So if we look here, that's this gene. So maybe we'll keep that in mind as we go forward. Next, we have this reverse transcription control. So this is the strand synthesis efficiency. You can see that all the QC wells on all 15 samples are below this threshold. So that's good. For the genomic DNA, we have a threshold of 35. If the well was had no value, it means that there was such low contamination that it couldn't, it didn't amplify at all. So we're looking for all the bars to be above this line or absent, which they are. All right, and then if you go to this processing summary, it just has a little bit more information on some summary statistics for the data that we uploaded. But other than the one housekeeping gene, this all looks good, and I'll show you how you can deal with that on the next page. So here's the data normalization page. There's 3 options here. Which wells to remove For example, usually we would remove all of the QC wells because we're probably not interested in knowing whether they're differentially expressed. But we can also check for outliers. So genes that have outlier values. And you can look at the FAQs for more details on how outliers are defined. So we'll leave this to remove all QC types. Next is what to do with non-detects. So when there are missing values in the data that you uploaded. Here we're going to impute non-detects. So that just means that we're going to replace missing values with some reasonable estimate of low expression. Now there are two methods for normalization. The main one is delta CT, where we use housekeeping genes, and then the second one, quantile normalization. This was developed initially for analyzing microarray or RNA-seq, but it's increasingly being used for CT arrays as those have been getting larger. So here we're going to stick with delta CT normalization, but we remember that this B2M gene was differentially expressed, so we're going to remove that. And we're going to make sure that our control group is the control. So now we'll update that. And we'll submit these changes. All right, so now we can look at our data. We can see PC plot of the samples. This is good. We're seeing pretty good separation across the different conditions. Some density plot and some processing summary. So we might notice there's this one sample, this high sample, that seems to be a little distinct. It could be an outlier, H1 here. But it's not too bad, and it's in the high dose, so it's possible it could just be biological variation, so we're going to leave that in. But if you wanted to remove it, you could go back to the quality check page, and there is a data editor button that allows you to remove certain samples. And it's on the quality check page because if there was one sample that was failing all of the quality check controls, you'd probably want to remove that before you proceed to normalization and differential analysis. All right, so next we have the differential expression page. There's two different ways of doing differential expression analysis. ANOVA makes some assumptions about normality, whereas Kruskal-Wallis is fully non-parametric. We're going to stick with ANOVA. We only had one type of metadata, the dose, so we don't have to change this. But if you had some other descriptors uploaded with your data, like sex or time or something like that, you could choose which one you wanted to use for differential expression analysis. And then we're going to use a common control. So we're going to compare all of our dose groups, our medium and our high dose group, to the control. There are more advanced options down here if you have a more complicated experimental design, and you can read about those in the FAQs for more details, but typically we can just ignore those. So I'll submit that and then proceed. So now we see a selected comparison, control versus high. We've put a p-value threshold of 0.05, and we are not filtering by log2 fold change, so we've put this at 0. So if we look, there's 61 were significantly upregulated and 29 were downregulated. And if we look at our table here, you can see the ones in green are downregulated, the ones in red are upregulated. And if we go to a much later one, we would see that maybe they're not significant anymore. There's also an AOP table. So we've gone to the AOP wiki and pulled out all of the pathways that have genes in them, and we've highlighted any that have a gene that was significantly differentially expressed. If you flip through, you can see the other ones. See, these ones weren't significant, so they're not highlighted. And if you are to click on an AOP or on a key event, it'll pull up the page on the AOP wiki where you can get more information on that pathway. If we want to look at the results for medium, we can switch it and resubmit. So you see now it's updated to a different number. And in general, the fold changes are much, much lower here. All right, so I'm going to go back to high. And then I'm going to proceed. So next we have EcoTox module summary of the results. Submit this. So we're looking at the high exposure group compared to the control. And what we're doing is we're taking the median log2 fold change of each of these different pathways. If the value is greater than 1.5, we're coloring it yellow, and if it's greater than 2, we're coloring it red. So these thresholds are just arbitrary right now, but in the future we hope to base them off of some previously measured samples. So these are these EcoTox processes, where there's just 5 larger categories. Then we also have the EcoTox modules. So this is 21 separate categories, and so you can get an idea for what biological processes are most impacted. And then there's also a result table with all of these in a little bit more detail. All right, next we'll go to visual analytics. And so this page, I'm not going to go into these in much depth, but these are interactive visualizations that you can look through all of your results in more detail. I'll just go to one as an example. So here, these are those same EcoTox module results, but now you see this is interactive. So if you click on one, it'll put all of the genes that make up that module into this box so you can see the individual results. And then if we click on one, you can see their violin plots down here. So this just makes it easy to kind of go through and view all the results. I'll leave the other ones for you to explore on your own, and good luck with your analysis.